Okay, so let's talk about replication of DNA. When we talk about replication of DNA, one of the things we have to keep in mind is that each of the strand of DNA is anti-parallel in that they do not run together. Now that means that there is a polarity to the actual DNA backbone or the DNA strands. One side will always be in the what we call the 5' position, the other side 3' position, and so on. So we'll take that up in just at the end of this discussion. The nucleotides that form the DNA molecule have their bases facing inward. Again, something we will look at in a little bit later. And so as they face each other, they are attracted to each other by hydrogen bonding. This hydrogen bonding needs to be reduced so that there is a separation of the DNA double strand to make what's known as a replication fork. With the fork in place, the nucleotides can now be allowed to slip into place and line up with a appropriate nucleotide nucleic base. So for example, if there's adenine here, it will wind up with a thymine base nucleotide. If there's a thymine here, it will end up with a adenine based nucleotide, like so. Once that is complete, endo and exonucleases will come by like editors and make sure that the nucleotides are in fact in their proper place. If not, they can actually be removed. The endonucleases can remove one nucleotide at a time. The exonucleases can remove 100 at a time. After that is done, ligase action will allow for the individual nucleotides to be connected, but connected in terms of linkages, chemical linkages, which will then allow for a firm backbone which has been created brand new. So what you wind up with is two DNA molecules, each with the original strand of DNA and each with a strand created de novo or brand new. Now, in terms of this business about anti-parallel, let's take a look at that, see if we can make some sense of it. Let's see if we can build a DNA molecule from scratch. So what I have here is a 5-carbon sugar called deoxyribose. The difference between ribose and deoxyribose is like one oxygen atom. Okay, so if we're doing biochemistry, yes, we would go into more picky details, but at this point I want you to understand the concept of the anti-parallel backbone, because that's an important situation, especially when it comes to replication, Okazaki fragments, etc. The structure here is oxygen. Here's carbon number 1, carbon number 2, 3, 4, and 5. So your carbon number 5 is right here. Your carbon number 3 is right here. Okay? Now, this continues on. So this will always be carbon number 3 on this side. There'll always be carbon number 5 on this side, right here. Okay? Now, if I were to take this situation and go ahead and make sort of copies of it, in effect, grow the strand, create, essentially a polymer. And what I'll do is I'll go ahead and make it a lot smaller so I can add more, so you can get a full effect. Go ahead and reduce it by 50%. See? Now I can go ahead and add some more. And you can see that no matter what I do, the number 5 position will always belong up here somewhere. The number 3 position will always belong down here somewhere with regards to the carbon on the deoxyribose molecule. So basically, we've established a polarity for this strand. What about the other strand? So let's take a look at the other strand that we're going to make. So I'm going to have to carefully, well, looks like I'm not going to be able to do it this way. So I'm going to have to take the whole thing. There we go. And by simply pasting it, you can see, very easily that while it looks parallel, the nucleotides are facing in the opposite direction. I mean, they kind of, well, they're facing not in the opposite direction, they're facing the same way, but they should be facing each other, right? So in order to make them face each other, let me try to flip this thing horizontally. And you can see that this is not going to work either. While a few of them might sort of face each other, most of them are missing each other and not even anywhere close to linking with each other. So I'm going to have to flip this one more time. Only this time I'm going to do it vertically. And you can see the magic happen as I flip it vertically. They're all lined up. All the nucleotide are lined up. The nucleic bases are lined up. So now adenine will be attracted to thymine. Cytosine will be attracted to guanine. In effect, they'll be like married listings, right? They're sort of gonna only link up with each other. Again, these are hydrogen bonds. They're not covalent bonds, because if they were covalent bonds, it would be almost impossible to replicate DNA. So that's why we got the hydrogen bonding. And as a result, you now have a anti-parallel set of strands in the DNA molecule. Okay? Let me kind of pan this a little bit so you can see the effect.